Welcome to this explainer. You know, it's pretty wild when you stop and think about it. You can literally chop a tiny piece off a houseplant, stick it in some dirt, and boom, you've grown a completely new, identical plant. But obviously, you absolutely cannot do that with an animal. So today, we're diving into the fascinating world of cloning to figure out exactly why that is and how science has somehow managed to bypass those natural limits to achieve the impossible.
We've got a great roadmap for today. First, we'll look at the magic of micropropagation, followed by its pros and cons. Then, we'll dive into the famous Dolly method for cloning mammals, weigh the risks and rewards of mammalian cloning, and finally, explore the transgenic medical future.
Section 1. The magic of micropropagation. Let's head straight into the plant lab to establish our baseline. This process is called micropropagation or tissue culture, and it happens completely in vitro, meaning outside a living organism, usually in a sterile dish. It's honestly a bit like following a very strict scientific recipe. First, you take tiny cuttings from a plant, those are your explants, and you thoroughly sterilize them. Once they're totally clean, you pop them into a nutrient-rich agar jelly. That environment makes the cells multiply into this blob-like mass called a callus. Finally, you add a specific mix of plant hormones, and that callus sprouts right into a brand new little plantlet. It really is an incredible journey. You're starting with just a simple leaf cutting taken with a scalpel, giving it a sterile wash, and setting it on an agar plate. From that one tiny explant, it multiplies into a callus, and before you know it, you've got a whole glass house packed with perfectly identical cloned plants, ready to go outside. It's just a brilliant process. Section 2. Micropropagation, pros and cons. So why go through all this trouble? Well, for commercial agriculture, the upside is massive. We can turn out genetically identical plants cheaply and in absolute huge quantities. Plus, it's fast. These clones mature way quicker than seeds, and you can grow them any time of year, completely ignoring the weather outside. Because it's all done in a sterile lab, they're completely disease-free. And hey, if the parent plant was naturally resistant to a certain bug, every single clone gets that exact same superpower. Conservationists love this too, because it's a real game changer for stepping in and saving rare endangered plant species. But, and this is a big but, it's a massive double-edged sword. First off, you need highly trained people and specialized sterile labs, so it's not exactly easy to just set up in your garage. The real biological danger, though, is the total lack of genetic variation. Since every single plant is a literal clone, they share the exact same weaknesses. Just one new pest or a sudden shift in the environment could theoretically wipe out the entire crop in one go because they just don't have the genetic diversity to adapt. And that brings us to the ultimate biological pivot point. Why is this so easy with plants but impossible with animals? It all comes down to a superpower called cellular differentiation. Plant cells keep the ability to transform into any specialized cell type for their entire lives. Animal cells, not so much. Once an animal develops, its cells are locked in. You can't just take a piece of animal tissue and grow a whole new individual from it. The only exception is stem cells and developing embryos. Once the animal matures, that magic window slams shut forever. Section 3. The Dolly method. Cloning mammals. Okay, so if we can't just take a skin cutting to clone a mammal, how on earth did a team in Scotland manage to clone the very first mammal back in 1996? They actually bypassed that entire stem cell hurdle using something called adult cell cloning. Here's the blueprint. First, you take an unfertilized egg cell and hollow it out by removing its nucleus. Next, you extract the fully formed diploid nucleus from an adult body cell, like a skin cell, and drop it into that hollowed out egg. Then you hit it with a tiny electric shock to jumpstart cell division, form an embryo, and place that embryo into a surrogate mother. Think about it like a biological heist. You've got sheep A, the one we actually want to clone. We just need a standard body cell from her. Then you've got sheep B, who is just providing the raw hardware, an empty, unfertilized egg. The scientists extract the vital diploid nucleus from sheep A's cell and completely toss out the nucleus from sheep B's egg. The real magic happens right at the merger. That diploid nucleus from sheep A, which holds her entire genetic blueprint, is carefully inserted into sheep B's completely empty egg. Then zap, a tiny electric shock tricks the cell into starting mitosis. It starts dividing and growing right into an embryo. That resulting embryo now contains the exact 100% genetic code of sheep A alone. It gets transferred into third sheep, the surrogate, who carries the pregnancy to term. The lamb that's born shares absolutely none of the surrogate's DNA. It is a perfect genetic clone of sheep A. And that, folks, is how we got Dolly the sheep.
Section 4. Mammalian cloning, risks and rewards.
With Dolly proving that mammalian cloning isn't just science fiction anymore, we have to talk about the massive global implications here. We are balancing some mind-blowing potential against very real biological concerns. On one hand, we could potentially save critically endangered species, or, crazy enough, maybe even resurrect extinct ones. In agriculture, farmers could quickly produce huge quantities of their absolute best, highest quality livestock. But the risks are basically the same as our cloned plants: zero genetic diversity. The populations become incredibly vulnerable to diseases. Plus, the data shows that cloned animals often just aren't as healthy as naturally bred ones.
And when you start talking about applying this to humans, the ethical alarm bells start ringing loud. A huge part of this stems directly from the biology itself. Cloning in humans has an unacceptably high failure rate. When it fails, it can result in stillborn children or children with severe, heartbreaking disabilities. It's a very stark reminder of the intense responsibilities that come with playing with this kind of biological fire.
Section 5. The transgenic medical future. Now, despite all the hurdles and risks of cloning everyday animals, the core technology is absolutely pushing the boundaries of human medicine, especially when we combine it with genetic engineering. This brings us to transgenic animals. Simply put, a transgenic animal is one where scientists have intentionally inserted a foreign gene directly into its genome. We are literally editing the genetic code to get a very specific result. Why go to all that trouble? Well, imagine an animal that can naturally produce life-saving human medicines right in its milk, its eggs, or its blood. Because that inserted gene can be designed to code for a highly useful protein, they can do exactly that. Once scientists successfully create just one of these animals, they can use those cloning techniques we just talked about to reliably copy it over and over again. This isn't just theory either. It's currently being used to manufacture vital proteins to treat human diseases. Which leaves us with a pretty heavy question. We are essentially weighing this incredible ability to manufacture life-saving human medicines against a severe lack of genetic diversity, genuine animal health issues, and some massive ethical dilemmas. So, considering the high failure rates and vulnerable gene pools, is the medical promise of cloned transgenic animals actually worth the risk? It's a massive, complex question right at the frontier of biology. Thanks for joining this explainer, and keep questioning the science around you.